All right, so we're going to finish out this
Actually, it didn't make any sense. Yes, it no, it didn't. So much sense. Say it again.
All right, so we were talking yesterday about the immune system and specifically about how there is an explosion, a clonal explosion of T cells and B cells that occur when a professional antigen-presenting cell, like a dendritic cell or monocyte, there's a couple of the other ones, that move their way into the lymphatic system, that kind of filter system that's connected to the circulatory system. And we have a bunch of these nodes. And as we talked about yesterday, when you have an infection, okay, doctors will feel for them. And there's a couple of different places. And the sinal cavity we have also is a place for the lymphatic system. And we have a bunch of important nodes in our top of our area because this is where we have most of the openings of our body. This is where we get a head cold, a chest cold, bronchitis. Okay, so there's a lot of infections. We have mucous membranes and open, soft, wet tissues that are basically nothing more really than cultures for bacteria if there was no immune system. So we have these warm, wet places with plenty of food and for cultures of microbes to grow. Now, that explosion of T cells and B cells, when there is an antigen, okay, present that a T cell can essentially identify, but it doesn't identify, as we talked about last week, the entire antigen. It only can identify a piece, and there's a reason behind that. That reason is the T cell wants to be specific, and if it had to identify the entire antigen, I don't think we can make that many copies of T cells that can identify exactly the antigens that we need to see. So what we do is, of course, a professional cell takes in, okay, the antigen, chops it up, presents that antigen, okay, on its Mhc proteins. We have the class 1 and the class 2. What were the difference?
Class 1 just kind of shows them, and class 2 can identify.
Well, the class 1 is...
No, class 1 is the protein that identifies the cell as part of the body?
Yeah, that's basically the one that identifies, it's the identifier if the cell's okay. If you're not under attack, viral, bacterial, or any kind of microbial attack or infection, you're going to have your indicator with nothing on it, and that tells all the other cells that you're fine and you're one of us. And that's also the part of the cell that we look for, like natural killers. They come by, which are, they're lymphocytes, but they're part of the what? The adaptive immune system, because they basically go around looking for the MHC1, looking for what? Looking for some piece of the antigen being on them if you're under attack. So natural killers, cells, NKSs, come around and say, oh, your MHC1 is different because you've been infected, you put a piece of the protein there, time to kill you, okay? But that cell just goes around and looks at every cell. Not a great thing if you've got a lot of cells. Yes, Frank?
About the bacteria in our gut, how do we tell the ours? Because they're not exactly ours, right? They're more foreign.
Well, it's a symbiotic relationship, and that's the symbiotic relationship. Remember, the weirdest thing is our gut is not really our inside. When you think about it, it's an open tube in our body. So what does the gut do? It transfers information from the outside in. So believe it or not, our small intestines is really the outside of the body.
Wait, really?
Yeah.
So wait, there's a hole going.
But they're inside our body.
The whole thing?
Yes, they're inside our body, but they're really on the outside. It's only when we digest them that they go into our what? Cells. It's an opening that stuff can travel through. So even though we have an opening, that really is not the inside of our body. So we can have bacteria on the outside. It's when that bacteria crosses a cell membrane into other cells and gets into our bloodstream. That's when we're transferring cancer cells or other things. So if it stays on the outside, our bloods don't go outside our body. If that was the case, you'd see blood jumping through our skin. So I know it's a weird concept, but remember the germ layers? Okay, remember the germ layers we talked about? The ecto, endo, and mesoderm? Okay, well, this was the what? This is the mesoderm. This was the what? Ecto. And ecto is where the skin is. So this folded over. Okay, so it folds over, and then you've got this lining, basically the tube, which is our digestive tract. So I know it's a weird concept. It's a great question. Thank you.
The difference between natural killers and cytotoxic T cells, is that just the response?
The difference is one is clearly the adaptive. Cytotoxic T cells are activated by a T helper cell that has found, just by rote chance of making all these different, basically receptors, just by chance in cutting up DNA, we found one that matches, activates it, throws out cytokines, okay, and all of a sudden you have all this humoral, we have the response of these cells, the cell-mediated response. Natural killer cells, although are lymphocytes, meaning they do spend time a lot in the lymph, which is that filtering system that connects to the blood, they're part of the innate. They go around checking Mhc basically proteins to see if there's something on them that's foreign, and if it is, it's killer. It just kills them. That can't be an adaptive response because that looks at every cell it can see. As you can see, it probably can't get to them all. So if you're under a huge attack, your innate system is going to get overrun. And that's why if you lose your T cells, like the HIV virus, a cold can kill you. Okay, because we get overrun very quickly when the whole body's under attack. When we're fighting infection in local areas, we can stop that. But when it becomes a whole body attack, where now the bacteria is in our blood and everywhere else, there's just not enough macrophages and monocytes, eosinophils and neutrophils to deal with them. We need to have something that goes after just those cells because there's so much of us, and that's why the adaptive had to evolve for bigger creatures like us, vertebrates. Yes?
So if you just have like a giant cut, say all over here, and it was infected, would it be overrun?
Well, there are infections that you can get from a cut that become dangerous and gangrene, and you could die from that infection.
The basic ones, like the ones that our innate system can take care of, but what if that gets overrun?
Well, then if it gets overrun, then we have to have adaptive take in to stop that. Sometimes the innate takes time, doesn't it? We measure the innate by the antibodies that are produced by the plasma cells, which are the effector cells once a B cell gets activated. So we measure the innate by how much antibodies for that particular antigen are being made. As you can see, it takes 14, 20 days to get a peak of antibodies after this. So you may die in that time period before we produce enough antibodies to kill that. So affection like a cut can be very serious really quickly because our primary immune response takes a while to kick in. And that is why, okay, you need to get, in that case, a what? A doctor and an antibiotic. Okay? To wait for this to occur might be too long. You may lose limbs and you can lose your life. Okay? So sometimes these are fast and furious types of infections that we just can't wait. Now, we talked about vaccines. Okay, why do we take vaccines? And there's a lot of things in the news that say, "Oh, I'm not vaccinating my children. It's dangerous." And there's some poor data out there. There was a study, and I don't remember it exactly, but as we know, we've been doing a lot of work with looking at research, and I believe the study had at most 10 children in it. Okay, and taking 10 people, you know the population number is small. There could be a lot of chance that occurs with 10 people outside of the factors you're studying. So out of the 10 people who had the vaccines, okay, 2 became autistic children. And what happened in this study, now again, I'm trying to recreate the data in my mind, I don't know exactly, but it's something with a small number of children, and they became autistic. So the researcher, based on his study, said, well, there may be a good chance that these immunizations, these vaccines, are causing this type of disorder, if you want to call it a disorder, autism in children. that these vaccinations are partly to blame. And you have a lot of people who have autistic children who deadly believe that, okay, and trying to clear their blood out of these, quote, vaccines that they had. Or some people are choosing not to vaccinate, okay? And we are seeing in the last 10 years an uptick in deaths of certain diseases that we haven't seen in centuries. Smallpox. People are dying of smallpox. People haven't died of smallpox in big numbers since 1920s, 1930s. Okay? Measles, mumps. People are dying. If you get measles, there's a 1 out of 1,000 chance that you can have this pressure of the brain. It's a disorder where your brain swells and it's life-threatening. So measles can be deadly. So people by either third world, even first world, are starting not to vaccinate their kids thinking that those things are harmful, when in fact they're an easy lifesaver. And what are vaccines, as we talked about before? Vaccines are usually a dead form of the antigen that just has what? It has the receptors still. If you take in a dead bacteria or virus, you're still taking in the receptors. The receptors get hit by chance, a B cell or an antigen-presenting cell that brings it to a T cell who, what? Identifies with it. And of course, we start the adaptive immune system that we talked about yesterday. We have this proliferation of B and T cells in our nodes, and eventually we have the effector cells of the B cells, plasma, that make the antibodies. And of course, we have the cytotoxic T cells, the cell-mediated response that go after the antigen throughout the body. But the problem with that is it takes time. So the idea behind a vaccine, and you have to know this, although it takes time to build up the immunity, it's the secondary immune response that's going to be quick and deadly. So if you were to get measles, okay, there's a lot of time before what? You build up some immunity. And during that time, you could have symptoms that are life-threatening and die, especially if you're a young baby, okay, or an elderly person, or even a healthy person. Maybe this is too long. But with a vaccine, exposing them and producing what? When there's a proliferation of B and T in your lymph nodes, what we learned, there's also proliferation of the memory T cells and B cells. So maybe you have 3 out of 10 billion cells that would attach to any potential antigen that comes in. Now you have, instead of 1 billion, now you have maybe 1 million that are sitting in your lymph nodes waiting to respond pretty quickly. And they do. There's no waiting period. It's a very quick response. So this, by having a vaccine, makes the response very quick. So there's no chance of having those deadly symptoms. But people by the millions are not vaccinating their kids with the thought that they're dangerous. It's dangerous not to. Crazy. Yes?
Wouldn't it, if you get autism, wouldn't that have to do something to do with like your genes?
Right. I mean, we think that, and I don't know if those genes have been, I don't think they figured that out exactly, but when you have this information that people spin and it's out there, people, and again, I do not have autistic kids, okay, so it's easy for me to sit here and say it's not this, not that, but when it's close to home, people want to believe what? Anything, okay, and I'm not saying these people are not intelligent, but you want to believe there's a cure, okay, because at this point, you know, those things are what they are, but yeah. Okay, again, easy to have simple, an easy mind about things, but it doesn't, it's not close home to you. Yeah.
About what you said, how like smallpox is coming back. How is it if if we already have a vaccination? Is it
because there's many people that are not choosing not to vaccinate their children, so the disease is going to spread and has killed people, has killed kids, or has made them disabled because of the symptoms. When in fact, it's preventable. Expose them to a dead virus. Expose them to the receptors so you build up antibodies. So that if you were exposed to it, boom, okay, those memory T and Bs explode upon contact and produce all the antibodies that get rid of them without them causing any harm. Why wouldn't you do that? Okay? Yes?
Would this result in dead diseases such as polio to come back?
Polio is coming back. Smallpox is coming back. And we know something which is really sad is those diseases are not mutating. Their receptors have been the same for 80, 90 years. The flu virus, influenza, okay, that is constantly changing. So you may be vaccinated on a dead virus strain. You may not get that strain. That strain could have mutated so quickly that you could still get sick. You're just preventing that basic strain from getting you again. So there are viruses or microorganisms that we get sick from that mutate so fast that it's hard to vaccinate against. But these things, polio, mumps, measles, all these things, that basically is a cocktail for my kids in terms of when they get a shot, are all preventable. And people are dying because on the basis they might be harmful. And so it's actually it's a big topic, and it's very sad that those things are coming back. But you need to understand why. So this is what we talked about yesterday, but this is understand the second response, the secondary immune response is because of the memory B and T cells that are ready to be activated, that are moving within your lymph. Yes?
How do they receive the dead antigens for the medicine they get?
Well, they basically, and I don't know exactly, but somehow they take a dead cell and somehow basically put it in some kind of solution that's probably isotonic with your blood, and basically you're shooting it out, probably maybe lysed cells that just have the cell membrane. Somehow that you have to have the receptors there to create an adaptive immune. You have to have the receptors for T and B cells too, or a professional antigen presenting cell to engulf and present for a T cell, I should say. Okay, because T cells, I don't want to say this, T cells don't directly attach themselves to antigens. B cells can, and B cells can produce the antigen itself. T cells need a professional antigen presenting cell. If I misspoke, I'm sorry. Okay, now, this is also important when you think about pregnancy. Okay, you have a baby and you have the mother. Okay, the baby and the mother, okay, are two different beings. Okay, and while the baby is in the womb, okay, there's placenta, and antibodies of the mother can travel across the placenta. The placenta is basically this tissue of the mother, okay, that connects the fetus and the baby. So we know that there's the blood of the mother, the blood of the baby, but there's a diffusion process of material through the placenta, and antibodies from the mother can travel and do so to the baby to give it its adaptive or some form of immunity to anything that it can run across. So when a baby is born, it is born with the antibodies of the mother, full complement. But those antibodies are proteins that do degrade. Yes?
Can antigens also pass through the placenta like that?
Yes. Yes, and that's what we're going to talk about the RH factor in a second. Yes.
Couldn't they take the antibodies that the mom has and the diseases she's already had and bring them to the baby so that they would have a secondary immune response?
No, because the antibodies are the result of B cells the mother has that have been activated. So the mother has the memory cells or the B cells that exploded at one point. So the antibodies are not an antigen. So when you have antibodies running through you, we're not going to identify them as antigens. Okay, they're just proteins running around. They're not cells. They're not cell receptors. So it's a good question. So no. So over a time period, the baby, okay, once it's born, starts losing its adaptive immunity that it had from its mother. Yes?
Is it possible to take a vaccination and then have those antibodies travel to the baby?
I believe that they do so, but I don't think the baby has the developed B or T cells yet to make that adaptive immune system. Since they're developing and need the mother, I do not believe their bone marrow is developed enough to be making B cells or T cells yet. So those antigens or those antibodies, those things, that baby can't produce them themselves yet.
If you vaccinate the mother.
If you vaccinate the mother, what does that mean? You're vaccinating the mother so you stimulate B and T cells, right, of the mother to produce the adaptive immunity. The baby doesn't have developed enough bone marrow to produce B or T cells yet. I don't know when that starts happening, but it doesn't happen in the womb. So vaccinations, which are just antigens, right? The body doesn't know they're dead or unharmful. It just, hey, that's a receptor we don't like. The baby doesn't have that ability yet, developmentally. And so that's the reason why that doesn't happen. So when the baby's born, it starts, the antibodies it had that crossed the placenta are now degrading and it's losing its ability. However, very interesting enough, when a baby is breastfed, okay, through the lactate of the milk of the mom, antibodies are what? Transferred. So among the nutrients that breast milk gives to the growing baby as a mammal, we're also transferring more antibodies until the baby builds up its immune system. So very healthy, not just for nutrients, but for antibodies when babies are breastfed. If you don't breastfeed your baby, okay, they're more susceptible to all of these diseases early on. So there's a health aspect to that as well. Yes?
Then, if babies are like fed formulas rather than like breastfed,
Yes.
wouldn't the formulas also have like antibodies?
No. Formulas cannot have any antibodies. Antibodies are what? Proteins that are made from B cells. Okay? So the formula is just basically some kind of sugar or carbohydrate they put in a certain way that's easily digested by the baby, that's full of vitamins, but there's no antibodies. There's no B cells producing them. Okay? We haven't, as far as I understood, taking B cells out of bodies and start making them antibodies and putting in the formula. That's not happening. And if it was, that'd be formulas that everyone would breastfeed their baby because they couldn't afford that. Okay? Yes.
Is that the only thing, the difference between breastfeeding and formula?
No, I think there's other documented things, but you know, we're a different society now. Mammals have always been based upon the idea that the mother rears the babies, and so evolutionary mammals, mother stayed with the young and raised them, and there was a definite benefit of producing milk for the babies. For instance, babies are always hungry. If you had to hunt every time a baby was hungry, you'd run out. So you always produced milk. You had milk available in their crazy feeding and sleeping times. So it's an evolutionary response to that. Now, of course, now in modern age, what are we doing? We got to go back to work soon. We don't have time for this, okay? We have the convenience. But that convenience leads to susceptibility of diseases for the early baby until their immune system picks in. And of course, my kids, we went back to work. My kids were breastfed until at least, I think my first kid, didn't want to stop. I think he was like 3, and the other one did for a year. But they both right away went to daycare. And some people keep their kids in from daycare, like they do their own. And our kids always went daycare, and they got sick right away. But now they never get sick. They're exposed to all those antigens early on. Their B and T cells created those memory cells. And now my oldest, I think, has been sick for school one day out of, he's in 5th grade now. So out of 6 years of school, he's been sick one day. And I know people that have sent their kids, kept them home until kindergarten, and they're sick all the time. So it's interesting. Now, of course, there's more to it than that, and people have different genes and different types of immune systems as well. So, but it's interesting when you look at that.
So then how can like some people like get sick once every year or so from like a common cold if they already have?
Well, the common cold is a virus, and viruses have a unique ability to mutate very, very quickly. So therefore, you have the cold, by the time you get another cold, it's a different strain. So you need a different receptor that we don't have a memory for, so we have to go through the what? The process.
So that's why it lasts so long.
Exactly. But you're not going to have the same cold twice. All right? Now, we're talking about blood, okay, talking about placenta and baby. Let's move on to blood types, okay? Blood typing. Now, we mentioned this before in genetics a little bit, but now we're ready to talk about the universal donor, okay, and all the things we've been talking about. So essentially, there's 4 blood typings, okay? There's the A, there's the B, there's the AB, and there's the O. Now, what does this mean? And this is pretty cool. Now that we've done all this, we're kind of ready for the explanation. Yes?
Don't each type has its own type of RH protein?
The RH factor, the rhesus factor, is the positive or negative. Okay, we're going to get to that.
Yeah, but
If you are an A blood cell, okay, you have a blood cell that produces a certain carbohydrate, and we'll call that an A factor. So this is a carbohydrate that's specific. It's a polysaccharide that comes off of the cell membrane. Here we go again. It's the same idea. The B cell has a carbohydrate. We call it the B carbohydrate. It's a little bit different. I don't know if it's chained or not. Okay, I'll just make it a little different. The AB, okay, has both. Has both the A carbohydrate and it has the B carbohydrate. And the O has none. Okay, I'm O. Now, what's important about blood typing is that you can't mix and match blood typing. You're going to have catastrophic effects because of the immune system. These are essentially receptors. Now, if you are a type A, this is really cool, all right? You are going to produce antibodies for B. Well, wait a minute. Why would we produce antibodies for B when we're A? Because we produce B cells that have what? And T cells that just by chance of DNA replication and cutting up and resplicing, we have what? Receptors for any possible what? Encounter. So it isn't like we definitely make antibodies for what? B. It's just that we have so many receptors already stored in our lymphatic system and regular circulatory system that if you were to give me B blood, we would see that blood as foreign, produce antibodies, okay, and probably have a memory cell already ready to produce antibodies, and those antibodies would coagulate the blood, and you don't want blood to clump. When blood clumps, it clogs. When it clogs, that gives you a heart attack if it's in your heart, a stroke in your an aneurysm. You want blood to flow, among other things. It causes pressure in different areas. Okay, if there's a lot of it, it's almost certain death. Okay, deadly. So why do we have antibody Bs? The reason why we have antibodies for any possible what? That we make B and T cells randomly for any possible encounter. That's the beauty of it. So the B is also a possibility, a strong possibility, a definite possibility. Now, here's what's really cool. Why don't we make antibody A's if we have all the possible combinations? And this is the coolest part. Yes?
You can't kill your own.
Right. So if you make a T cell or a B cell, okay, more so a T cell, okay, it goes to your thymus. What do we talk about? The reason why it's called a T cell is because that white blood cell goes to your what? Thymus, and it gets processed. And one of the processing in the thymus gland on top of your heart is that it basically tests to see if it attacks itself. If a T cell made randomly by cutting up the protein and having these receptors, okay, if these attack themselves, they go cell apoptosis, they kill it. So before they release the T cells into the lymphatic system, they test it amongst their own what? Body. So we have this mechanism in our system where we kill the lymphocytes that tag our own body. Okay? So we kill off, if we did have antibody or T cells that would attach to this, okay, we kill them off because it's us. So we don't produce that. Yes?
So how does that work? Do you produce the proteins that are on the cell in?
How does it work is the same work is that we would identify, if you're an A person, A-type blood, and you gave me a B, we would identify this as not ours. So a natural-born killer would kill it, but not only that, probably not a natural-born killer because that's not an Mhc protein, probably would identify it as not ours. We would elicit a response by the B cells to produce plasma cells, to produce antibodies, and it would be certain coagulation. Remember, what antibodies do is they attach and clump them up. Agglutination is the word.
I meant in development.
What's that?
I meant in development and testing. Would they test the proteins that are on the molecule?
Oh, it's a great question. I don't know the answer, but somehow in the thymus, before they let the T lymphocyte go, that's the T's for the thymus, they see if it can identify itself as an antigen. If it does, they kill that one off. And there's going to be ones because we make the receptors totally randomly. I can't see. To me, it's hard for me to believe that we make these receptors totally randomly, and so many of them that we've got receptors waiting for things that we don't know exist. That's how many random hits that we have. But we do know this: if it's made of us, we won't attack itself because we'll kill that T cell. We won't let it proliferate. That's important. So the B cell is going to have what? Antibodies for the what? Okay, the AB.
None.
It can't have. If it did, it attacks itself. So this has no antibodies. Okay? Now, yes?
Do we produce antibodies for antigens that don't even exist?
Yes. No, we produce B cell and T cells for antigens that don't exist yet.
So we-
That is so spooky to me, too. It's crazy. We just take these receptors and we just randomly cut them up into different forms. There's a billions of them until a what? An antigen-presenting cell cuts them up in smaller pieces and puts them on their Mhc protein. And if that's the reason why we cut them up. Think about it. Why do we cut them up and present them? So that little piece is a good chance that one of those pieces can be detected by a T cell randomly. It makes perfect sense when you think about it. Yes?
Would a mutation like a deletion or a substitution or even frame shift be completely okay in the genetic mixing up of receptors?
Yeah, and I think that's part of the process. I think that's totally part of the process. I think mostly, though, this is definitely just the spliceosomes doing their jobs. But certainly, it's a welcome change. And that's what we talked about. Genetic variability in mutations is not always a bad thing. In this area, I think it's helpful to have as many different combinations so that what? You have that one B cell or T cell that can what? See that foreign material. And it's crazy that you would think, my gosh, how can you make something without knowing what's there yet? And again, before I learned this, it's hard to actually grasp. Okay, so we have the O blood, who has what? Okay,
A and B.
he's going to have antibodies for A and B. Why?
Because it can only accept one.
Right, so it's going to have antibodies for both A and B. Now, when you think about this, it makes perfect sense, okay, because it doesn't have any of the receptors, so these two are going to be foreign to it, so it's going to allow the T cells to have these receptors and allow them to exist. Now, when you look at these, there's something called the universal donor. Which one is that? Oh, I'm the universal donor. All of these guys, including myself, can accept O. Oh! If you're from Brooklyn, okay? Now, why couldn't you accept O? Because you don't have any tags. You don't have any what? So I have antibody B, A, or none, but there's nothing to attach to. Now, obviously, O has receptors, but it doesn't have these carbohydrates as receptors, and so the immune system doesn't attack it. So if you're an O person, your blood is sought after. Once they hear that you're an O, okay? Hey, get more blood. Yes.
Then what's like the positive and negative?
I'll get to that. And of course, who's the universal recipient?
AB.
AB. Okay? Because it has what?
No.
Right. It doesn't have, it's never going to make any antibodies for A and B because that's how it exists. So it can accept O, okay? But it can accept B and A because, okay, these things won't trigger response because they already have them. If they did, that means something's wrong with your thymus. So that should make, if I was to tell you this before we did the immune system, you may get a piece of it, but hopefully like, oh yeah, yeah.
Since the AB blood type, it has both A and B's. If you like had a blood transfusion with like only type A blood, wouldn't it work only like halfway since it doesn't have?
Remember, this is the recipient. Okay, this person can receive. Okay, B.
Yeah, but would it work as well as if the recipient got blood type AB? Because like since.
Well, yeah, sure. AB can accept AB, but it can accept all types as well because it doesn't produce any antibodies for these different receptors because if it did, it would be killing its own cells, and that cannot be allowed in the hypothalamus. The T cells get what? Killed off. Cell-mediated death. There's a great example of that here. Yes?
Oh, me.
I'm cross-eyed. Now you can speak, Sarah. Sarah, speak! Sarah! I have to yell at you one time. Go ahead.
So A or B would not be able to accept AB because of the other one that's in there. Like,
Exactly.
It would be able to accept its own, but then the other
Right,
one
because
would
you're gonna have antibodies for the A receptor. So this, if this accepted AB, okay, you're going to make B cells or T cells that are going to reach that. This part's going to be foreign to it, and we're going to coagulate it, and certain problems right there. Yes.
I don't know who you were looking at. What is the most popular blood type?
Popular.
A.
Okay. Oh! Depends who you're talking about. Okay, if you're talking about a truck that comes to our school and wants blood,
No, like
okay,
talking about
talking about in a hospital,
Population.
okay,
No, like
who needs
people.
help?
Just people.
People in the world.
Oh, it's a great question. I think it's very evenly distributed.
It's all positive and A positive. I don't know what's.
But I don't know what the percentages are. I don't think it's not like 2/3 of the people are
A
okay.
designated 2/3.
What's that?
A designated 2/3.
Wait.
Go on Google.com.
I don't think they're as far apart as you think. I think they're very close. But I don't know exactly.
40% O positive, and it's like 29.
How would you know?
Is it really that much? I have to look that up.
I don't, all I know is that
Someone Google the percentage of people that are O, AB, and AB. Yes.
How does like as you're being like when you're in your mother's stomach, like how does your the cells or embryo decide what type of blood type you're gonna be?
Oh, that's genetics. That's pure genetics. So we have genes on a chromosome that you basically the two alleles came together, right? And you decided that. In fact, if you think about that, if you're AB, what kind of dominance do you think this is? Codominance. If you can be AB, you're going to express what? Both alleles. Yes, full circle. Hey!
Well, then wouldn't,
Thank you for that question.
wouldn't O be recessive?
Yes.
So then how's
That's why I'm wondering as well. Okay, O, I believe, is the broken gene. Okay? And this, of course, is two A's, two B's, both AB. Okay, so here is codominance. Yes.
But like,
I don't know for sure. He's saying that.
but if like, let's say like a mom was like type A and a dad was type B, since they're co-dominant, wouldn't the world be mostly AB then?
It's a great question. But you have two alleles, don't you?
Yeah.
So if you're heterozygous dominant, you're right, but people aren't. Okay? So yeah, exactly. So if you're an A and an O comes together, you'll be A, right?
Yeah.
Okay, so so you have to think about the parents have two alleles they do, and they have to what? And meiosis grab one of them.
If they're like both dominant, then you would expect.
Exactly. So now we're getting back to what? September again. See how great this course is? All right. We're in what? We're in almost April, right? And we're talking about September.
This may be a dumb question, but do
Summer, right?
AB blood cells have both receptors and some that have just A and some that have just B, or is it all both?
No, they have both. They're going to have both. Every single blood cell will have both.
All right, we got the percentages. Do you want
Oh my
positives
God.
or just the total?
I just want total O's.
All right, so total O's is 45.
Wow.
Total A's is 40. Total B's is 11. And total AB's is 4%. So if you're
What?
AB, congratulations, you're part of the 4% of the population.
Okay.
Wow, Frank, that was so
Wait, so
nice.
then
there's also-
AB's co-recessive?
Yeah.
Alright, just make this more complicated. This don't.
Yeah,
You didn't
Yeah,
Raj
I
has
mean,
had
they've
enough
been
this year?
here
Yes.
mentally. He's been absent for most of the
This is
year.
also only an American population.
Oh, well, that's also.
Oh, we wanted the world.
Okay.
population.
Any case, what is that Rh factor? Because you can be O positive, O negative. They call this the rhesus factor. Okay, so it's called the Rh factor, and basically it's a protein that's on the outside. It's a different receptor on the blood cell as well. Either you have it or don't. Now, if you're positive for the Rh factor, okay, well, there's no better, but there's a scenario with the womb and pregnancy, okay? So I'll talk about that when you get back from break.
Part 2.